I welcome you all to the first orientation session for postgraduates for Omics Logic, Amity University, Uttar Pradesh Collaborative Training. We recently oriented the graduates for Omics Logic Training and Research, and now we are excited to orient you all and bring you the benefit for the collaboration between Amity University, Noida and Pine Biotech, where the objectives of this collaboration is to increase the awareness of biology as data science to address the urgent need to encourage, train, and mentor people to leverage available public domain datasets and tools to explore the world of biology. My name is Sonalika Ray, and I'm the Omics Logic Community Manager. I'm responsible for daily interaction with thousands of users that are a part of our bioinformatics community that is managed by Pine Biotech. So, before we begin with today's talk, I would first of all like to introduce us. We are a US based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy to use analytical tools, and our mission is to make bioinformatics more accessible. Omics Logic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. We are pleased to announce that we also recently collaborated with Amity University Jaipur, Chandigarh Group of Colleges Mohali, MIT Pune, and FABA, Federation of Asian Biotech Associations. Omics Logic training and research is running internationally in 8 different regions and over 15,000 users around the world. Due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. Omics Logic programs follow a project based learning approach, which use research grade tools to analyze data from top peer reviewed journals. Omics Logic training has been completed by participants from 162 countries in over 300 workshops for 6 different specialization tracks, being oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Omics Logic training and research uh, sorry for the glitch. I'm honored to have been working with a team that collectively has an amazing experience in this area, including the other co-founders of the company. Dr. Alfred Tauber, who comes from a medical background with an expertise in oncology, immunology, and biochemistry of information. And one of his major projects is the establishment of Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a hard work by some of the people mentioned here on the right. Julia Panner, who is an expert bioinformatician and helped prepare many of the materials that we would use. Avi Titovsky, who is a computer scientist and leads the development of the T-Bioinfo platform that we would use. Dr. Harpreet Kaur has an expertise in the application of statistical machine learning techniques on biomedical data and the development of prediction tools and databases. And she looks after the curriculum development, bioinformatics, and data science course development as well. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in peer reviewed journals and collaborations in industry and academia. Over the past several years, he has been developing collaborations with universities as a mentor and project lead for Omics Logic training and research fellowship. Dr. Raghavendra Lakshmi Narayan is an accomplished computational biologist with experience in large-scale analysis of multi-omics and data science methods in precision oncology and protein DNA structural bioinformatics. Applying his proficiency in using Python, R, and shell script for bioinformatics, Dr. Raghavendran is interested in translational approaches to understand cancer biology, neurological disorders, and plant bioinformatics. As students embark on their professional journey with freshly completed degrees, they often wonder how professional life will be different from studies. Many prepare for this moment in advance, exploring possibilities to work in an area of interest through internships and research fellowships during their studies. Amity University, Noida recognizes the need to enable students to participate in such meaningful experiences early on, leading to a pursuit of collaborations with industry. As a result, there is a natural fit between Amity University, Noida and Pine Biotech, a big data analytics company that is focused on bioinformatics and machine learning for biomedical data. As a result, Pine Biotech will work closely with Amity University Noida to develop a pipeline for students to get training and apply their bioinformatics skills in research and industry projects as interns and research fellows with the company. This process starts with a program designed to introduce students from all backgrounds to the intersection of biology and data science, and we call it Omics Logic. Now, to discuss about the collaboration and the programs, I would like to pass on the stage to our mentors. I would like to invite Dr. Mohit Mazumdar, who has completed his PhD in computational biology and has been working with academia and industry to solve important research problems. In his current role, he is working with students and faculty and scientists who have keen interest to explore biological data, to do research in bioinformatics, and provide guidance and training. Over to you, Dr. Mohit.
Thank you, Sonalika. And hello, everyone. It's really nice to see you all. Let me share my screen before I start my presentation. Okay, as people are still joining, I would just want to give another round of introduction, especially for those who are joining for the first time. So, my name is Mohit, and I have completed my PhD in computational biology. And I have been working with academia and industry for past several years on important research problems. And one of the reasons that we have collaborated with uh, Amity University, Noida, Uttar Pradesh, is the reason being is to be able to develop research projects and develop training for such research projects to be carried out by students in a group. So today I wanted to discuss a little bit about that. But before telling you about the process of how this is going to helpful in NTCC trainings and how this is going to be helpful in overall in your career, I just wanted to explain a little bit about my experience, how I got here. So I did my graduation in bioinformatics, and then I did my master's from Jamia and my PhD from Jawaharlal Nehru University. And then I went on to study at Cambridgeshire, EMBL, Welcome Sanger Institute. And then I also went to Canada University of Saskatchewan for research internships. So that was my academic career. And during that period of time, I was able to collaborate with a lot of faculties. But obviously, before that, when I was doing my undergraduation and my master's, I was not knowing so much about what is going on in the industry. I was not knowing much about the opportunities that are really there, because the important thing is that it is to understand that how the industry operates, how the research works, how the areas of life sciences and biotechnology works. So to knowing all of that, there's a need for industry academic collaboration, and there's a need for students to get exposed to industry. And that's the reason we are here. And I am hopeful that we'll be able to help you with all of that in the coming months, as we develop these relationships and develop research ideas and projects along the way. So to tell you about this, I think it was a historic memorandum of understanding that was signed between Amity University, Uttar Pradesh, and Pine Biotech, US, for joint research and trainings in the area of bioinformatics, data science, and computational biology. And I'm really, really glad to meet you all. I'm going to say it again because many of you are those researchers who would be collaborating with us and would be working with us and developing really exciting projects in several areas of research that you're going to hear about in the next few minutes, where you will be able to understand, train from a very basic level, and then get an understanding of developing research projects using a team of bioinformaticians and experts. So as I was telling you that this was a breakthrough because there was a lot of expertise and experts involved in such a collaboration. So we have over 50 faculties and over a group of technicians and experts from Tower Bioinformatics Research Center, University of Haifa, Israel. So Tower Bioinformatics Research Center, as explained by Sonalika, as you have seen, is a cutting edge bioinformatics research center. So we'll be obviously connected with Tower Bioinformatics Research Center, along with the faculties that we have in Amity University and the great researchers that we have at around the world collaborating through this Omics Logic research training. So this happened between several institutes. So Amity Institute of Biotechnology, Noida and Lucknow, Center for Medical Biotechnology, Center for Plant and Environmental Biotechnology, Center for Biotechnology and Biochemical Engineering, Center for Cellular and Molecular Biotechnology, Center for Computational Biology and Bioinformatics. So the discussions, the breakthroughs were done majorly by Professor Balvinder Shukla, Vice Chancellor, Amity, Uttar Pradesh, Professor Seema Bhatnagar, Assistant Director, Coordinator, NTCC, and Amity Institute of Biotechnology. So as a result of this collaboration, what we are expecting is that ability to provide high quality bioinformatics education and continue to expand with a wide range of sophisticated tools for new students who are entering this dynamic environment of data science and bioinformatics. And that's why we are meeting with you all and we are discussing this great opportunity that I think all of you are going to appreciate. So as a result of training, it is not just training, it is training and research and developing skill sets which are really, really relevant to today's world. That includes, finding collaborators amongst faculty, amongst mentors, and amongst other student groups that we have that I'll be introducing you very soon. So the first part, today we are meeting for the orientation, and the first part is to understand that what are the different resources that we have. And today we are going to, in the next stage of this presentation, Mr. Nalika is going to help you with that. So we are going to review basic concepts of genetics, basic concepts of biotechnology, and how modern biology is now being evolved from all of that, and then to develop clear basic concepts of bioinformatics and data science. What are the different keywords that are going to be used? What are the different keywords that you would see that industry and research are putting up for the job opportunities, for the internship opportunities, and for going for higher studies in world top universities like Harvard, Cambridge, MIT, and many other great universities that are there around the West and also in the East. So the thing is that we are going to do is in this orientation part, we would want to learn about some of the basic things that are going to build up to that different stages that we discussed, where we are going to learn about the technology, where we're going to learn about the application of this technology, and then when we are going to talk about how do you do it yourself, how can you become a master of this. So this also involves like application of R and Python that are really used in today's world for analysis of this big data, and then how to use these tools and algorithms in a systematic manner, in a meaningful manner, so that you can come up with solutions our work on problems such as drug discovery and uh, drug designing using machine learning, deep learning, and all the cheminformatics tools that are available out there. So the collaboration as such is for the Omics Logic Research Fellowship, and this research fellowship is a great opportunity to build a research ecosystem between academia and industry, where we are going to address challenges that can be addressed through your skill sets that you have developed as a biotechnologist, as a data, as a understanding how biotechnology is now moving to become data science, biomedical data science, and all of that. So today we are going to use some of the resources that we have put together for this innovative training, and that involves a couple of platforms. One is learnomicslogic.com that you can see on the screen. Another is server Bioinfo platform. So the learnomicslogic.com that we are going to explore in some time helps us to understand um, how do you like understand the basic concepts of biology, and then builds up to different advanced concepts and modern concepts, where we are talking about different type of data types that are generated from these experiments in the wet lab. And then how do you analyze that data using this powerful multi-omics platform, which is the server Bioinfo platform, where you will be able to run the experiments on the cloud by using lots and lots of data. So we are talking about big data, so we are talking about data in megabytes and gigabytes. So one of the reasons, again, I'm coming back to the reasons that why we are doing this. So, the collaboration between industry and academia is key to catalyzing innovation and growth. So industry partnership is instrumental in advancing research and in creating a skilled workforce. So in this era of growing research, cutting edge technologies have provided life science researchers to study this overwhelming data at a molecular level. And that is why it is very important to understand this that the spike in this data, which is like spike in genomic data, spike in gene expression data, spike in proteomic data, metabolomic data, and data sequencing, DNA sequencing data, which is the genomic data, has an enormous potential, and an enormous potential to solve some of the most complicated challenges, where life science and computers act as a balancing bridge in providing solutions to the scientific questions. And that's the reason we are providing this Omics Logic Research Fellowship. So as I said, you'll be utilizing some of these resources, and then there are different steps. So there are different steps, which includes basics, which is the topics that those are the topics that we are going to cover during the basic training, where we are going to learn from the resources that are put online, that you can take on your own convenient time and do it at your own pace. And then we have a lot of recorded videos by experts, short videos that explains a certain experiment, that explains a certain method that you can obviously learn from. And then we are planning several live sessions, hands-on sessions, on-site, online sessions, to help you go through these different steps, because we understand that everyone are coming from a different background, everyone has a different research interest. So we are providing this opportunity to be a more of an individualistic, personalized help to develop research topics that interest you the most. And then there's a feedback system that we have prepared in collaboration with your faculty and the researchers who are there at the Topper Bioinformatics Research Center, and also like the university collaborators and our team in the US, and also in different areas of the different locations around the world. So obviously there are different stages leading to different outcomes. So we'll be discussing them later. So what are the different advantages that we are going to talk about? Is I'd already discussed self-paced learning and the big data platform and this project-based curriculum. And then we have the expert mentors, and we have different collaborators from around the world. Like one of the collaborators that Sonalika talked about is the FABA. FABA is an organization where we have collaborated to design in collaboration with industry experts coming from big pharma and coming from AI driven drug discovery companies, to collaborate with us and enable and give that knowledge to the student so that they can understand that what is going on and understand that how drug discovery or chemoinformatics and biomedical data analysis, all of this makes sense with the advent of technology and with the solutions that are they're providing to the customers. And those customers are all of us, because I think healthcare and all the drugs that have been developed by pharmaceutical companies and other companies are being utilized by many of us. So that is about that. And then we have the platform that we are going to discuss. So there are a few things in today's presentation. So I'll be quickly going through about the different topics that you can choose from and become specialized in. And then we will have short presentations by Dr. Harpreet and Dr. Raghavendra, who are here with us to discuss about the projects we would be working on. And then, as I was telling you, that why we are doing this, because yes, biology is now a little bit different than what it was before, because now we are not doing single experiments, we are doing high throughput experiments. And when a system which is capable of doing high throughput experiments, then the amount of information that you get from these high throughput experiments becomes really, really big. So that information is one of the major challenge for anyone to interpret from, because data generation, as you see, is becoming easier and easier. Load a sample from any environment and put it in the machine, and most of the things are done automatically and you get the data. But once you get the data, I mean, that's where the challenge starts. So that's why biology has now characteristics of data science, and that's why this is relevant. So not just one, two, three, four, five, but billions of data points that are just there on genes, billions of proteins expressing in different organisms all around and at different time points. And this information, this information is compiled in large files that are available online on public repositories. And that's where we are going to utilize that information and make the most by doing some really in-depth scientific research, applying some of the algorithms, applying some of the techniques that are there right now, used by scientists all around the globe, and understand human health, understand living organisms, understand biotechnology, understand crop, understand livestock, work on oncology, infectious diseases, neuroscience, agriculture, and all those fields. So some of the keywords that you need to learn about, that you must have heard about, of course, are these simple keywords, like learning about omics technology, DNA sequencing and DNA sequences is genomics. You all have heard about ATGC. You know how ATGC are a string that runs, those lines are so long that a human genome becomes a 3.2 billion base pair long genome. Similarly, like transcriptomics, protein structures, these are all interconnected, and the understanding of all those important components are nowadays are going giving us a clear picture of how the system looks. So I'm talking about system biology here. I'm talking about problems that are associated with system, and with all of these things that we are seeing around. So I'm talking about variants, I'm talking about mutations, I'm talking about viruses, I'm talking about pandemic, I'm talking about the effect. So all of that is like related when we are looking for solution strategies, when we are looking for techniques that can help us do efficient drug designing, or at least help the community by providing right kind of results that can enable them to come up with such a discovery, to come up with such an intervention that actually is going to help humankind. So that involves utilizing this data, analyzing this data using techniques such as, machine learning and deep learning. So we are learning about this different type of omics data that contains a very detailed information of genetic information. Sorry about the last slide. So yeah, it contains a very detailed information about the genetic information. And what we don't realize when we learn about and hear about these techniques is that, okay, this is a technique and we get to know about this information. So what? You can ask that, like, what is different, like how this is changing things. This is changing things because when I'm talking genomics, it is like a complete map of all the DNA that is there, like entire map of all the DNA that is there in the organism. When I'm talking about transcriptomics, epigenomics, it is about the entire gene expression profile of an organism at a given point of time. So 20,000, 30,000 gene expression you're looking, and then you have a chance to compare. And when you do comparison, uh, yeah, as I was telling you, that when you do comparison, then there's this probability, to work or understand different types. So one person is having cancer, one person does not have cancer. You do the comparison, you see what is out there, what is different, and you come up with some new thing. So that's how it is. I mean, nowadays we do a lot of comparison. We're comparing the data that is available with the reference data, and then we are understanding so many different things. So that is a lot about bioinformatics, that how bioinformatics is playing a key role by analyzing the data, comparing data, and this is really doable now. So after getting oriented about all of this basics, like what are the different data types, what are the different technologies, and what are the different applications, like getting an overview, the next step is to learn about some of these technologies. So you can choose, like if you want to do, like if you're interested in, like for example, cancer, and finding mutations that are causing the cancer, then one of the technology that you can use is genomics, right? Similarly, there are different technologies for different research questions that can be answered using that. So the data that is being generated, the information that that data is giving, is something that you can discuss with the experts right here. All of us are capable of answering such questions, that what this technology can be applied for. Is it capable of answering your question? Whether there are data sets available to answer such questions, all of that. So I'm talking about these different technologies. In the next step, after you get oriented, you will be learning about these different technologies. And how you are going to learn about these different technologies? You are going to learn and experience the hands-on. So you will be seeing the data types, you'll be seeing the information, and then you'll be practicing on these different data types and understanding that how you can apply this technologies without need to learn coding. So we have a platform that is an AI guided platform, which you'll be seeing very soon, that would not require you to do coding. But once you understand the logic, that how it works, how this entire uh, analysis pathway goes, what are the different workflows that are there, you can then you can definitely, if you are interested, you can learn coding as well, because the platform is capable of handling Python, R, and doing downstream analysis. So if you are interested in coding, you can go for coding. But if you are not familiar, or if you don't want to do coding, then also you can do all the analysis and do good biological interpretation out of your data by that. But yeah, we would recommend you to because it's like connected, and it's very easy to do, because we already have everything prepared for you. So we would recommend you to go one step to another step, and explore all the resources that are out there, because you never know what interests you more, what interests you most, and maybe you'll find them really nice to carry forward with your skill sets. So the reason we are doing this is because there's a huge need, there's a huge gap, and there's a lot of demand for people who have data analysis skill sets in US. When you apply for a technical position, or when you are done biotechnology, or just applying technology. And when you have data science skill sets, there is a 2.5% higher, like fold higher salary that you will be applicable to get when you have both the skill sets. So you data science skill sets are really adding to that part. And similar to the that of different industry, so research industry is where you can apply this. Hospitals are using this, pharma is using this for sure. And after you learn about all of that, the next step is to learn about different specializations that are out there. So you can learn on different data types that we have and examples that we have prepared for you, that involves all of these projects that are available on the portal for you to take online. And this can be done through the steps that Sonalika will show you very soon. So now, once you have learned about the introduction, once you have gotten into the understanding of, you know, what are these technologies, how they can be applied on, you have learned it by applying it on example data sets. So you learned on, how the Ebola outbreak happened and what analysis we could do for understanding the outbreak that happened in 2015, 2016 in West Africa. That example was there, which I showed you in the last, presentation, last slide. But once you have learned it, like, you can think about the questions that you can answer. You can think about the questions that your faculty is working on, and how you can help your faculty to complete some of the analysis that he's planning to do, and maybe come out with a publication. So those are the real opportunities that we are going to present to you, that are going to help you enhance your profile, enhance your curriculum, enhance your CV, so that you can showcase that. And obviously we have an online platform that actually showcases all of that, that could be seen by anyone who is interested in hiring you. So the future of medicine, I was coming to the specialization. The future of medicine is precision medicine. And we discussed in length about why precision medicine is important. And today, in today's talk, Dr. Raghavendran is going to talk about his research on precision medicine, and how the different type of other omics data is actually enabling scientists to understand how this can be useful for individual, at an individual level. So precision medicine is an emerging approach for disease treatment and prevention that takes into account uh, about account of individual variability. The variability could be at a genetic level, at your in your genes. So genetically I can be different from you, because there's specific difference in our gene and the environment that we lived in. So today I'm little under the weather, and one of the reason is I should say not under the weather, I should say under the pollution. So pollution, I'm in Delhi, so there's a lot of pollution here, and I'm having allergies right now. So this is the environment that we are living in, that has an effect on our body, right? To consider that environment is to understand that how this, how you can sample some of the data that is there in your gut. So metagenomic data can be used to understand that how it is interacting, our gut microbiota is interacting. So all of that, there is a gut project, gut microbiome project that happened in the US, that you're going to learn when you learn about metagenomics. But going back to, precision medicine, it is important to learn this, because it gives you understanding of basic and translational research, that could be in oncology, that could be in infectious diseases, neuroscience, and especially in cancer. There's a lot of work that has been going on, especially because it's a very complicated disease, and we all know the repercussion of what happens when a person is being diagnosed with cancer. So bioinformatics is a huge plus in today's world for finding solutions, especially for precision, when we are talking about different type of data analysis that happens at different level. So when you start a drug discovery pipeline, you start by testing your drug on patient-derived cell lines. First, you do it on cell lines, then you do it on xenograft mouse models, where the tumor is implanted, where Dr. Harpreet is going to talk a little bit about this in her presentation today, and then how you do it on a clinical level to get that information. So at different stages, you will find data, and then you can see that how this is, working for different companies, and how some of the drugs got approved through this process by FDA. So one of the reasons that cancer can be, helpful by us, by bioinformaticians who can help our data analysts like all of us here who is going to work on these sort of challenges, is to help them by early diagnosis, by getting, um, by finding new biomarkers or signatures, molecular signatures for early detection and prognosis. So also, like, because cancer is a disease of aging, the more we're growing old, the more the chances are there for such mutations to happen. So if we can properly screen and early diagnose, we'll be able to save a lot of people. So I was going to tell you about one short case study. So this is that case study. And then we have EMR studies. And similarly, in infectious diseases, there's a lot of opportunity. Our team is working on, finding genomic sequences, differences in the genomic variants, and host immune response. So if you have any of the infectious disease project in your mind, you can obviously think about that and can work on such topics. Similarly, there are different topics within infectious diseases, and I've already told you about metagenomics, that how metagenomics plays an important role in understanding the natural environment, dissecting these communities, exploring species richness, and then finding those microbial community for human health and disease. And then we have exciting opportunities where we are integrating all of this data and finding that what is the outcome of such studies, what is the best that we can get. And similarly, after learning about all this, you have an opportunity to do work with a project which is driven more towards drug discovery, more towards, finding drugs by using this information that you got by the omics data, by using, maybe some of the faculty have already found some drug, found some target which can be, which could be a druggable target. So we can obviously plan to find targets and find noble drugs by involving hit discovery, and do some optimizations using AI. Similarly, we are going to work on finding new protein structures, which can be like protein, not new protein structures, but finding structures that we can, fold and predict using maybe Google's DeepMind, the software that they came up with in the 14th CRISPR critical assessment of structural prediction competition. So these are some of the opportunities to, explore, maybe do some alpha fold prediction, compare it with some other molecule, or do complex prediction such as that. So we are going to be discussing and doing research work in all of these areas, and we welcome you all to join and be a part of this opportunity, and get started with an orientation, and get, really familiarized with such an interesting areas of study that are available for you to take and undergo this sort of training and develop research project using the Omics Logic Research Fellowship. So that involves space omics, that involves, learning about astronomers health, that involves, working on complex problem. And all of this is simplified. All of this is really what we have been trying to do, is to simplify all of this and make it in a way that anyone, absolutely anyone, without a background of bioinformatics, without a background of, computational coding languages, can do this on their own. So obviously, I would like to welcome you all again, and really appreciate all of you joining today, and great to see you all. And if you have any questions for me, please put it in the chat. And for the first orientation meeting, now I would like to ask in Snalika to take you forward to the Learn login and show you the advantages of building a profile and going through the initial coursework as a part of this orientation. Followed by that, we will be able to meet also Dr. Harpreet and Dr. Raghavendran for their project information. Thank you.
Thank you so much, Dr. Mohit. So now I would like to take you all to the very same landing page where you all registered yourself. So while registering yourself, you might have noticed that on the very same landing page, you can see a step-wise process that we have tried to streamline to get you all started with the training and then move ahead with all of your research projects, right? So now let's get started. You all need to visit the Omics Logic Learn portal and sign up. Now, once I say sign up, you all need to create a free sign up account. So let's do this together. I would be giving let's a 5 minutes time to you all to sign up. Here is the link you may find in the chat box. Now, once you have signed up on the portal, please let me know in the chat box itself by sending in a one or yes, maybe, so that I know that we can proceed forward. It is very important to create an account so that you can get started with the training. Okay, so now let me do this with you. Either you can create your credentials, just type in your mail ID, create a password for yourself and sign in, or you can also sign up by your social accounts. It could be your Google account or Facebook account or anyone that you would like to connect with. So let me just sign up and show it to you. Please put in the chat box once you have completed your sign up. So as we are running short of time, I will now just quickly take you ahead, and others can sign up and also look at whatever guidelines are being instructed right now. So once you have completed your sign up, you are requested to please update your profile. Now, having an updated profile means that you must have a profile picture. Your name should be in title case because this is the same font that is going to be reflected in your certificates. You must connect your social accounts and also add a brief bio about yourself so that we know what is your background and what are your research interests. And all the courses that you would be taking on the OmicsLogic Learn portal, you can see all of them here under the courses tab on your profile itself. And once you complete the courses, you might be able to download a certificate as well from the profile tab itself. So I just wanted to give you a very quick overview of one of your fellows profile, whose name is Dhaval Kumar Srivastava. Dhaval is an MTech Biotech student, and I would like to highlight his profile just to showcase you what score he has and how beautifully he has updated his profile. So as you can see, Dhaval has all his social accounts linked and his name is in title case, and he also has a brief bio about himself. He did
Hi
complete a lot of
Sonalika.
courses.
Hi Sonalika. I think you need to change the screen, your screen.
Oh, okay, ma'am. Thank you so much. So this is the profile I was talking about, Dhaval Kumar Srivastava. As you can see that all the courses that he completed are visible in his profile, and also the project that he would be submitting very soon. He has submitted the project in his college, so he would be uploading the same on the profile too. And also he has already downloaded and shared a lot of his certificates on the social media platforms. Now, why is it important to have a complete profile with all the details I mentioned? It is important because we do feature profiles based on their progress. So having in all the things updated is going to help you share your all your updates on social media. And also with each and every course that you will complete on the portal, you are going to get some points. And as you can see, Thawal has already scored 19,000 points and has reached an expert level. Now, let me get you all started. So once you have registered yourself and updated your profile with all the details that I have mentioned, the next step is to complete the basic training. So before we start off, with a with a research fellowship program, or before we start off with your own independent research project, it's very important to be well versed with the concepts that are required to carry out that research project, to know what the concept is, to know what the methodology is, to know what the algorithms are, how they're involved, and to get some hands-on practice by running the pipelines. Also, Dr. Harpreet would help you all to run those pipelines, and she will also give a glimpse of one of those in today's session. So now, once you have completed your profile, then comes the basic training part, where you need to complete the very basic 3 courses. The first one is Introduction to Bioinformatics. The second one is Bytes and Molecules. And the third one is how to design a bioinformatics research project, right? So let's just go to these courses very quickly and see what's in here. Also, this is your first assignment. So you all are supposed to simply register yourself, complete your profile, and just complete these first 2 courses that I just mentioned: Introduction to Bioinformatics and Bytes and Molecules. And you must be able to complete Introduction to Bioinformatics within 1 week, and the Bytes and Molecules could be covered within the second week. So we are hoping that you all must be able to submit your first assignment by the end of the second week. So let's see what's an Introduction to Bioinformatics. In this particular course, you will be able to understand the basics of bioinformatics. So bioinformatics is not at all a different field altogether, but it is actually a field that is integrated these days in all the various domains of biology, be it biotechnology, microbiology, pharmacy, biochemistry. You will find some element of bioinformatics in the field, definitely. So it's important to understand what is
Sorry,
bioinformatics, why
Sonalika, to
is it
interrupt you.
important.
Sonalika,
And
we have already explained this in detail, why bioinformatics important. So I think let's move on to the tasks and then pass it on to.
right, sir. Thank you so
Yeah.
much, sir. And the second course is Bytes and Molecules. And this particular course is going to help you understand the various domains. What sir talked about: genomics, transcriptomics, metagenomics. So here you will find all the introductory lessons where you can just understand the basics and then proceed further with whatever specialization you might want to proceed with. Okay, so these are the 2 courses that you have to finish, and also make sure that you have a complete profile. And just after this session, I would be mailing you out a form which has to be submitted by the end of second week, where you have to just submit your Learn New ID. Now, what is a Learn New ID? This is your Learn User ID, and we can just monitor your progress again and make sure that you have completed the assignment just by visiting your profile ID. So please make sure that you submit your assignment here. Now I would like to pass on the stage to Dr. Harpreet. Over to you, ma'am.
Thank you, Sonalika. And thank you, Dr. Mohit, giving the overview about bioinformatics and this program. Okay, so now let me share my slide. Hello, everyone. So myself, Harpreet, and I have a PhD in bioinformatics. In my current role with Pine Biotech, I'm curriculum developer and research consultant. So where I'm mentoring research fellows and working with international collaborators in various research projects based on biomedical and GS data. So already Dr. Mohit has explained you and gave you overview about different omics fields and their application. Now let me give you a glimpse how bioinformatics can be used on biomedical data to drive significance meaning by taking some example projects so that I had completed during my PhD and completed with number of research fellows like you. So before we start working on any project, we need to understand the background of that research question. So my PhD was in the area of cancer genomics, specifically liver cancer. So when I went through the literature to understand what are the major loopholes associated with this malignancy. So I have observed liver cancer is often a misdiagnosed and detected too late for an effective treatment. Another factor that contributes to the poor outcome of the liver cancer patient is the frequent reoccurrence of the tumor after the treatment. So with keeping these concerns in mind, so I have addressed some of the possible research questions. So in the first project, we use large-scale transcriptomics data to identify diagnostic biomarker panel for a primary liver malignancy, that is hepatocellular carcinoma. So in this study, we use 30 publicly available a dataset containing nearly 4,000 samples to elucidate transcriptomics biomarker panel for diagnosis and prognosis of liver cancer, implementing various data mining techniques. So in the second project, we focused on another liver malignancy, that is cholangiocarcinoma. So in this study, we used 9 datasets containing 350 cholangiocarcinoma, 133 Edison non-tumors, 90 hepatocellular carcinoma patient sample to scrutinize cholangiocarcinoma specific transcriptomic signatures, implementing various machine learning techniques like extra-tree, SVM, random forest, logistic regression, etc. So in the next project, we focused on identifying the biomarker that can predict liver cancer at an early stage. So in this project, we use transcriptomics and methylation data to identify a biomarker panel for stage classification of SCC. So here we use 350 samples and eventually elucidate 51 feature-based biomarkers, which include 30 RNA transcripts and 20 methylation CPG sites using various data mining and statistical methods. So in the last project, we focused on identifying multi-omics signatures that can stratify low occurrence risk group and high reoccurrence risk group of the SCC patients. Here we identify 90 RNA, 50 miRNA, 50 methylation, and 4 integrative features. So besides, I have worked with number of the research fellows with Pine Biotech. So many of these research fellows, they don't have much skills in the bioinformatics or any programming language. So how they able to implement different skills and what they come up with? So let me give you some of the glimpse of the project that were completed by our research fellows. So first we have Miss Simay Dulanar, who is an undergraduate student from Turkey, completed her project entitled Long Non-Coding RNA as a therapeutic target in chronic lymphocytic leukemia using publicly available data and various tools that are integrated on T-Bioinfo server that Dr. Mohit was talking about, and I will also demonstrate it to you. Then Ms. Urja Parekh has completed her research project identification of biomarker to differentiate between neural stem cell and glioblastoma stem cell, employing publicly available data and using various bioinformatics methods. So then apart from the oncology, then we have another research fellow who worked in the field of neuroscience. So here Ms. Rutvi Vaja completed her project entitled In Silico Analysis of Transcriptomic Profiling an Affected Biological Pathway in Multiple Sclerosis. So then another very important field that Dr. Mohit was talking about is space omics. So then we have a research fellow, Ms. Dilara Dekane, who has completed her research project in the field of space omics. Particularly, she worked on a project entitled Radiation Threat to Human in Space, an alternative approach with probiotic using metagenomics data and various bioinformatics tools that are integrated on T-Bioinfo server. So apart from neuroscience and oncology, then I have also worked in the field of infectious disease with the research fellows like Mr. Supanthate has completed his project in the field of malaria. in specifically analysis of gene expression profile to study malaria vaccine dose efficacy and immune response modulation using transcriptomics data of the patient using various data mining techniques. So apart from the transcriptomics, we also work on genomics data with Ghana team. So then beside graduate and postgraduate or PhD students, then there are some of the research fellows who are just high school students. So like we have Ms. Netra Srinivasan, who has completed her project entitled Effect of Onset of Menopause on the Transcriptomic Profile of Aging Female Brains. And then Mr. Andres has completed his project in the field of Alzheimer's disease, particularly where he studied amyloid beta induced neutrophil activation may provide a link between the two pathological hallmarks of Alzheimer's disease condition. So then we also worked with the research fellow from Amity University as well. So let me just take you to directly to the on the Learn platform where you can find their projects as well directly. So let me just first share link with you. So they are also worked in various fields, including infectious disease, oncology, and neuroscience. So for instance, Ms. Kanuva Sharma here completed her project on bioinformatics analysis on small cell lung cancer patient with bone metastasis using publicly available data. Then Ms. Akanksha Singh completed her project on in silico analysis of NGS data of glioblastoma to understand the impact of IDH1 mutation. And you can also go through these videos to understand exactly what they had done. So then we have Mr. Aditya Vajpayee completed his project on transcription analysis of HIV infection. Then Ms. Shruti Goyal completed her project on transcription analysis of early detection of cervical cancer. Then Mr. Haridev Thakur has completed his project on identification of biomarker for the prostate cancer. So far, we were talking about like how different research projects can be completed by using using publicly available data and implementing various bioinformatics techniques. So now let me give you the glimpse like how we can do it. So let me just demonstrate it to you. Let me take you to this T-Bioinfo server. So here we have different subsections. If you have look at NGS data, mass spectrometry, structural biology, then various data integration or data mining techniques. So then apart from that, we have also specialized tracks in the field of cancer biology, virology, genomics, and epigenomics and metagenomics. So depending upon your research interest, you can explore these tools. And of course, to understand how this pipeline and analysis tool works, you need to go through the basic courses which are associated with these kind of analysis. So now let me demonstrate to you by taking one example of Pdx project. So here we have how we can generate simple gene expression table from raw sequencing data. So if you look at this raw sequencing publicly available data, so we have like 128 sample where each file is like 4 GB, 8 GB or something like that. If you want to process this data on your own system, it is not possible. So how you can process this data itself by using these kind of platform here. So this particular platform will help you to understand this data, how to process this process and analyze that. So I'm taking example of this data itself. So where we have nearly 128 samples and from which we have taken like already selected desired 20 samples or so. So here I'm taking this Pdx project. So what is Pdx? So these are the patient-derived xenografts where a tumor, freshly tumor tissue is transplanted from human to the immunodeficient mice. So here we have the data from breast cancer, where we have two types of breast cancer, that is ER positive and triple negative. So to how we can process this data using this platform. So once because this is an example pipeline, so we already uploaded the data. So here you need not to do anything. You just can click on OK. So by clicking on OK, we will able to upload the data. Then here, different options automatically will get highlighted once you uploaded the data. To understand which step you need to execute, that explanation is given within the course, but right now here I'm just showing you, like here we have to first perform first step under the pre-processing. So here you need to understand, like this is the option that I need to perform and how and what this particular step is doing. So you will also get detail here as well. So when you click on the option, and for example, this is Trimomatic. So Trimomatic will remove adapters from your raw sequencing reads. So then the next step which is getting highlighted, that is Pcr Clean. So you need not to write any program. So you just need to do click an option. But before clicking on or using an option, you need to understand the logic. So while clicking on OK, I'm able to perform Pcr Clean, which will remove any Pcr duplicates from our data. So the next options which are getting highlighted here under the mapping, if you look at these algorithms, so they are under the mapping on genome or transcriptome. So here, as I just want to generate a ready to use expression dataset matrices, so I am performing mapping or alignment of of these reads on reference genome. So you will also get details here itself, like what this tool is doing. So then by clicking on OK, we will able to execute this step. Then once we able to perform alignment of reads on reference genome, next, how we can quantify that. So here you will, the option which is getting highlighted, that is RSCM. So this algorithm will eventually calculate the abundance of your reads again, for a particular gene or a particular transcript. So here we able to, by clicking on OK, we will able to execute this step. Then there is two options getting highlighted. So this is for differential expression analysis. Right now we can skip this, but I'm ending just my pipeline here. By clicking on it, we will able to complete the pipeline. So here you will able to obtain eventually these outputs. So then how to analyze these outputs, because you finally have clear gene expression table. So those things you will learn when you going through these different courses. So yeah, now if you have any question, please feel free to post your query in the chat box. And I'm looking forward to work with you. So yeah, thank you.
Thank you so much, Dr. Harpreet. Now, I would like to call Dr. Raghavendran to please share about his research project. Over to you, Dr. Raghavendran.
Thank you very much, Sonalika. Let me share my screen. Okay, that's about it. I'm sorry, I've gotten to the last slide. Let's start the first. Okay. Yeah, thank you, Sonalika, and thank you, Dr. Harpreet. Good afternoon to one and all. I'm Raghavendra, Bioinformatics mentor and research consultant with Omics Logic. Let me brief you about my background and research interests. I'm a computation biologist by training, and I specialize in subjects like multi-omics data analysis, data science methods in precision oncology, and protein and DNA structural bioinformatics. So I completed my formal education in bioinformatics and biophysics and graduated PhD in computational biology from Max Planck Institute, Germany. Then I worked on projects involving cancer genomics, plant bioinformatics, protein structural bioinformatics in the Institute of Bioinformatics and Applied Biotechnology, Bengaluru, and at the Jawaharlal University as a postdoctoral research scientist. So throughout my research career, I developed special interest and developed my expertise in precision oncology through data science methods applied to multi-omics data. So right from my PhD years, I have mentored several undergrad students, and I feel privileged to work with some of the bright young minds and also postgraduate students like you all in the past, also in the future. So I wish and I sincerely hope that you all enjoy as much as I do in this partnership between Amity Noida and Pine Biotech USA. So that was a very concise view of my background and research interests. Now I want to briefly speak about one of my research projects involving precision oncology. I chose to talk specifically about this study because it utilizes a very well established pipeline of multi-omics data analysis on publicly available data. So in the end, I hope this project will inspire you to develop your own scientific curiosity, leveraging the abundance of publicly available data in the platforms available. Okay, sorry for that. Okay. So we know that cancer is the leading cause of the disease deaths worldwide. So for such a dreadful disease, systematic cancer treatments involved or have relied on simple chemotherapeutic drugs that are marginally more toxic to cancer cells than normal cells. This is a huge problem because on an average, only 25% of the cancer patients respond to cancer drugs. In comparison, about 80% of the patients respond to popular painkillers available in the market. So a prominent solution to this problem is precision oncological therapy, where a treatment strategy is designed by targeting the molecular characteristics of the patient's tumor. Hence, the treatment is personalized to the patients who are likely to respond to specific cancer therapies. There is not sufficiently enough data from cancer patients to molecularly characterize tumors under multitude of different treatment options available at the market now. So hence, we have to resort to suitable model systems that was introduced to you earlier by Dr. Mohit and also Dr. Harpreet, like in cell line data and also in immunocompromised mouse data, which are also called as patient-derived xenografts. So this study involves data from patient-derived xenografts. So I have already covered why we have to do such a study, and let me go through how this is done. So the tumor cells from the cancer patients are subjected to next-gen sequencing techniques, and the information about the genetic expression and the gene mutation and copy number variation were extracted. This data molecularly characterizes the tumors. Then these tumor samples are then grafted into immunosuppressed mouse and administered several cancer drugs. The tumor's response to their respective drugs were documented as a tumor volume data and the mouse's survival response data. This data records the tumor's reaction to different treatments that were administered. So both the molecular characterization data and the treatment's response data are available along with the publication. Such a wealth of data generates a plethora and a lot of research questions. My specific interest using this data is to design and implement machine learning models to predict tumor responses to different treatments available. So what did I find? So I find the treatment responses predicted was more accurate when I use information from all the genes compared to information from single gene biomarkers that was traditionally used. So in the leftmost part of the graph, you see that this plot deals with a machine learning model that uses data only from the single gene biomarkers to predict the treatment response of all the tumors. So the second graph, it demonstrates you the accuracy which has to reach at MCC value of 1 to be a perfectly accurate machine learning model has improved significantly when I use the genetic mutation data from all the genes available. So furthermore, if I include more, let me go to this. Yes. So furthermore, if I include more omics data like copy number variation and genetic expression, gene expression data, so my consistency in the prediction accuracy improves leaps and bounds. As you can see, this spans a whole array of -1 to +1 with no consistency in prediction to when I use samples from all, when I use information from all the omics data and compared to the single gene biomarkers, I get a very consistent prediction around 0.5 and above, which is considered very good prediction for such a sparse data that is available. So the accuracy, hence I have just demonstrated you the accuracy of the prediction increased when more than one type of sequencing data were used to train the machine learning model. So what else could be done more in such a study? So with this list of most informative genes, we can extrapolate that into gene set enrichment analysis and try to identify specific biomarkers that explains the tumor's resistance or the tumor's sensitivity to given treatment. So one can extend also, in addition to that, one can extend such an analysis to include patient data from the TCGA repository. That would give us a sneak peek at the performance of machine learning models, which were built and trained on preclinical data like mouse immunocompromised mouse data and tested or but applied and tested on real world patient data like the TCGA patient data. And so these are all the interesting research questions that one can extend that this type of multi-omics data integration analysis into. Hope this was informative and inspiring for you all to generate interesting research questions from publicly available data. To perform such an analysis, coding is an integral part, but if one lacks coding experience, that should not deter you from asking such an interesting questions. And OmicsLogic's platform bridges that gap by providing the necessary training and platforms required you to perform such an impactful research. And let me demonstrate you. Why is this not working? Okay, great. So I want to provide you with a sample of how lessons involving coding are organized in the Omics Logic platform. These are some of the courses that you will find in the platform. And these courses that involve coding are organized and directed specifically towards students who have completed the introductory courses in their respective subjects. And all these courses have the following organization in the part involving executing R and Python courses. The organization includes what analysis are we doing, why are we doing such an analysis, and what do we expect to learn from doing such an analysis, and finally, how do we perform such an analysis. The codes, especially the R and the Python codes, themselves include comments at each step explaining the purpose, the syntaxes, and the variables. So it becomes very easy to follow through. And finally, the output generated from performing such an analysis and how is this output interpreted with respect to the biological questions in hand. So with that, I hope I would have inspired you to ask several interesting research questions despite lacking prior experience in coding or expertise in coding. And if you are an expert in coding, we have several options available to expand your skill and knowledge in the specific areas. And with that, I would like to conclude my talk and thank you all for your attention and hand over the podium to Ms. Sonalika.
Thank you so much, Dr. Raghavendra. And I would just like to thank everyone who joined the meeting and understood about the Omics Logic Amity University Training and Research Program. I would be now mailing out all the details to you all. It would be nice if you get started with your first assignment and be responsive by submitting your profile IDs. Thank you all once again for joining today's session.